Hey guys, what we're gonna do now is we're gonna go through two of these stages. I was gonna say all of them, but not. So we're gonna go through how does a cell do DNA replication. We're gonna do that first, and then later we're gonna talk about how does a cell divide. For G1 and G2, essentially a cell just gets bigger, and the way a cell gets bigger is it increases its surface area. And so growth factor will be released like we talked about over here, and that will signal the cell to start producing phospholipids, more cholesterol, more integral proteins, and then the cell will grow in size and also start making more organelles. So we're not going to really focus on these two stages. We're going to focus on the S phase and the M phase. And then for today, we're going to focus on the S phase. But first, you need to know what DNA looks like because we're going to replicate DNA. So you need to understand the structure of DNA. So flip to your next page. Let's title this DNA structure. Wonderful structure. You should know what this means, but it stands for deoxyribo nucleic acid. So deoxy is the D, ribonucleic acid. So if you remember way back to chapter 5, this is one of our macromolecules, is a nucleic acid. There's only two types, there's DNA and RNA, and we'll talk about RNA later in the year. So deoxyribonucleic acid, there's a few roots in here that'll kind of help us identify structures in this molecule. So the ribo part, the deoxyribo, is referring to the sugar of this molecule. So if you remember, all macromolecules are made up of monomers except for lipids. So deoxyribonucleic acid, this is our polymer. So nucleic acids can occur in this polymer form, or they can occur in the monomer form, which would be a nucleotide. Okay, so this is our polymer, and then we'll just write down here nucleotides are the monomer. So those are those single units. If we bond many of those units up together, you'll get the nucleic acid, the polymer. So the deoxyribo here is referring to the sugar of our nucleotide. All nucleotides are made of 3 parts. Let's draw it. They're all made up of a 5-carbon sugar. The 5-carbon sugar actually has an oxygen at its peak, so we're going to start with that oxygen, and then you're going to draw a pentagon. Yep. This is our sugar. Okay, sugar. The sugar is called, from our root here, deoxyribose. So this sugar is deoxyribose. It's a 5-carbon sugar, and we're going to count the carbons in a second. The second part to every single nucleic acid is bonded to that sugar. There is going to be a phosphate group, which is PO4 minus. So DNA is actually slightly negative, which makes it move in particular ways, which is super cool. The last part to every nucleotide, and this is the part that varies. So this is the section we call the backbone, just the sugar and the phosphate group. And remember, when we call something a backbone, we're assuming that it is the same throughout. So the backbone is the same for every single nucleotide. The part that differs is our last section. We're doing it in a different color, and this is our nitrogenous base. My box isn't big enough to write nitrogenous. I'm just going to write base. We say it's a nitrogenous base because it has nitrogen in it. They all have nitrogen in some form. So there are 4 bases. You should know these: A, T, C, and G. So you should know A is adenine. And if you remember ATP, adenosine triphosphate, it has an adenine. Remember GTP, guanine diphosphate, guanine triphosphate, cytosine, and thymine. Yeah. There are base pairing rules. They're called Chargaff's base pairing rules. So there is this man, his last name was Chargaff, and he discovered that particular bases will always pair with each other. So let's kind of back up a little. When we were discovering DNA, you should know, like, the famous scientists Watson and Crick. They actually stole some information from Rosalind Franklin, a female scientist, and she got credited for discovering the structure of DNA posthumously because she died discovering the structure of DNA. So what Rosalind Franklin did is she did X-ray crystallography. So they knew there was this like genetic material. They thought it was protein to start with, and then they started realizing this is something else. And so what Rosalind Franklin did is she took strands of DNA and she put them into crystals. Okay, so she kind of encased them in a crystal, and then she exposed those crystals to X-rays, right? So it's a electromagnetic wave. And when she's exposed the crystals to X-rays, when the little X-rays kind of hit the crystal, if there's anything solid there, like DNA, it'll diffract. So she ended up with this picture. It's called a diffraction pattern. But Rosalind Franklin is who discovered that DNA is double-stranded, so it's a double helical structure. And she died due to the radiation she was exposed to by the X-rays from her research, and she actually got, it was either uterine or ovarian cancer because it was, the X-rays were at that level of her waist, and she died. And then Watson and Crick took what she had discovered and claimed it as their own, and then they said that they discovered the double helix. Rude, I know. So Rosalind Franklin is the one who discovered that DNA is what we call double-stranded. So we're going to draw one strand. Cute, just make a wave, just a squiggle. And she discovered that it's double-stranded. So there is a second strand. Make another wiggle. Cute. Before she discovered this, it was thought that it was triple-stranded. People thought it was single-stranded. So without her, we wouldn't have understood DNA replication, protein synthesis. I am going on and on because female scientists need more recognition. Okay, my rant is over. DNA is said to be anti-parallel. The two strands are parallel, so it's like if you take a ladder and then you twist that ladder, the two backbones are going to be parallel to each other, but we say they're anti-parallel. And what the anti refers to is it refers to the directions of our DNA. So this kind of has to come back to this sugar. With this 5-carbon sugar, we actually number the carbons. And so when we're talking about the carbons, we can all be talking about the same carbon. So if I say like, oh yeah, on carbon 1, we all know what carbon 1 is. When you identify your carbons, remember in chemistry, whenever 2 lines junction, that means there's a carbon there. You always start clockwise from the oxygen. So here's my oxygen. I'm going to go clockwise. Here's my first junction. This is carbon number 1. I'm going to keep going clockwise. Here's my second junction, carbon number 2. Keep going clockwise. Next junction, carbon 3, carbon 4. Right? There's not going to be a carbon here because we don't have a junction. They told us there's an oxygen there. But on the bond between the phosphate group and the sugar, there is another carbon here, and that is our 5th carbon. So when we figure out the direction that nucleotides are facing in a DNA strand, we only focus on the number 5 carbon because it's the topmost carbon, or the one on this pole. I don't want to say topmost because it might not be on the top, but it's on this pole of our molecule. And then we focus on our 3 carbon because it's on this pole of our molecule. So now we have 2 carbons that are on opposite poles, one that's closer to the oxygen, one that's further from the oxygen. So when we label directionality on a strand of DNA, we label them from the 5 carbon end, and we call it the 5' end, and then just follow your squiggle all the way to the end to the 3' end. Meaning, on this purple strand of DNA, my nucleotides would be oriented like this, where the 5' carbon is closer to this portion of my paper, and the 3' carbon is facing this end of my paper. If I take this nucleotide and I flip it, like so, you can see that now my 3' carbon is more towards the top, and my 5' carbon is more towards the bottom. So it just gives us orientation. So if I was trying to figure out directionality for my green strand, my 5' carbon is more towards this side. Did I do that right? Oh no, we needed to flip. Sorry, this needs to be like flipped. I'll draw it. The 5' carbon will be down here. This is getting crazy. I wrote upside down. Okay, 5'. And then up here will be the 3' carbon, because our nucleotide is going to be mirrored and flipped. We're going to draw it. Okay. So one strand will always go from 5 to 3', and the other will be anti-parallel to that. It will always run 3 to 5. You cannot have a DNA molecule that has the 5' bonded, because they would both be facing like this. So the bases, which I drew as these little rungs, wouldn't bond. If we want to have these bond, I need to take this strand and I need to flip it, and that makes my 3' end match up with my 5' end. Okay, so let's talk about Chargaff's base pairing rules. Okay, so here's how Chargaff discovered these base pairing rules. Our bases are all slightly different in size. Some of them are double ring structures and some of them are single ring structures. So let's kind of talk about that first. So we have a set of bases that we call the purines. And another set that we call the pyrimidines. Cool. Pyrimidines, almost like pyramids. Purines are all double ring structured bases. So these have double ring. So they're longer in size. Pyrimidines are all made of a single ring structure. So they look something like this. So the double ring structure would be something like so, where you have one ring and then you'll have another ring coming off of it. So they have a 2-ring structure, so they're longer. Pyrimidines are made of a single ring, so they're shorter. Like so. What Chargaff discovered is when he looked at... I'm not sure. Oh, creepy, my Alexa just went off. When he looked at the diffraction patterns of DNA once they started imaging it with Rosalind Franklin, okay, my rant is over, but she is importante. They found that the shape of DNA is uniform all throughout. Like the width of it doesn't change. Let me explain this. So when we looked at DNA, oh, my cat's whining now. You can kind of see that in a 2-dimensional structure, it looks like it dips in, but imagine that it's like 3-dimensional, right? And we're just twisting. And so the width of DNA is constant. So what Chargaff discovered, kind of looking at all these images of DNA, is like the width of DNA is staying constant. If the width of DNA is staying constant, that means you must always have a purine bound with a pyrimidine. Because if you have 2 purines bound together, it's going to make the width bigger because you'll have 4 rings total across. If you have 2 pyrimidines together, it'll make the width a lot shorter because you'll only have 2 rings across. So in order to get this uniformity, it must be a purine with a pyrimidine always, and then you'll always have 3 rings across. The purines are adenine and guanine. So this is our A and G. And then our pyrimidines are C-cytosine and thymine. Okay, so you'll always have one of the purines paired with one of the pyrimidines. So you should know the base pairing rules. Adenine will always pair with thymine, and they always pair with a double bond because they have 2 pairs of electrons they can share with each other. And then cytosine will always pair with guanine, and they have 3 pairs of electrons they will share together. Okay. The way I remember who bonds with who, C and G, they're both curvy. Curvy goes with curvy. A's and T's, very linear letters. Cool. I don't know if you'll need to remember which is which, but I went to Davis and the saying was, all Aggies, AG, are pure. That's how we remembered it. So all Aggies are pure. And that helped us remember that A and G are the purines. And then pyrimidines, C and T. That spell, this is a stretch, guys. C and T. Cat. Pyrimidines. It's so close to pyramids. Think of the Egyptians. What animal did they worship? Cats. C, T. Okay. That's how I remember it. Okay, we're gonna draw a strand of DNA. So we're gonna draw our individual nucleotides. I have this much room. I'm gonna try to fit 3 nucleotides on each side. Okay, we're gonna draw a double-stranded DNA. So we're gonna start out with, let's use the color you can see. Black. Do it. We're going to start with a nucleotide that has the 5' end up here, and then we're going to do nucleotides that have the 5' end down here. So we have our anti-parallel. I typically start with my sugar just because I like to start with my oxygens first. So here's my deoxyribose. Here's my phosphate group. Here's my nitrogenous base. Let's make this one cytosine. Cytosine. Cute. Let's draw another nucleotide right below. They all go in the same direction. If they're on the same side of the DNA strand, they're all in the same orientation, right? So like just a recap, if we need to number our carbons to figure out directionality, this is carbon 1, 2, 3, 4, and then your 5th carbon is up on that branch. You always start clockwise from your oxygen. Another helping hint is the oxygen always points to the 5' end because they're on the same side of the molecule, okay? So if you're not gonna remember this counting, just memorize that the oxygen always points to the 5' end. Always, 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 okay? Let's draw another nucleotide right below it. So you start with your oxygen, draw your pentagon. There's my deoxyribose. Here's my phosphate group. Let's do adenine. I think you know what word we're about to spell. And then let's draw one more nucleotide. Here's my oxygen. Here's my deoxyribose. Here's my phosphate group. I know, unpredictable. Cat. Cool. So we have 3 nucleotides, 3 monomers. They are not bonded together. So we're going to do dehydration synthesis, and we're going to bond our monomers together. A covalent bond will form, and that covalent bond forms between the phosphate group of one nucleotide and the sugar of the next nucleotide. It'll look like that. That was easy. Phosphate group of one, sugar of the next. So these are our new covalent bonds that are bonding our nucleotides now together. So now we have a polymer. It's single-stranded, but we now have a polymer nucleic acid. So this bond, it's a covalent bond. Generally strong, relatively strong. It's called, get excited, the phosphodiester bond. That's a mouthful. Okay, so we have 2 phosphodiester bonds in this diagram. Next, Let's figure out our directions. So we always have oxygens pointing towards 5', or you can look here, our 3' carbon is closer to this side of our strand. Our 3' would be here. The 5' is closer to this side of our strand. So we would have 5' up here, 3' down here. So that's our directionality for this strand. It just helps us orient ourselves. Now, when we're going to draw the opposing anti-parallel strand, if this is difficult for you, you can turn your notebook just upside down and like kind of do what we did here. But you just need to draw everything flipped and mirrored. That's exciting. So let's start with our bases. C pairs with G. Okay, so if I have a cytosine here, that means this nucleotide over here will be a guanine. So I'm just going to start with my base. It's bonded to my deoxyribose sugar, but now my sugar is going to be flipped, okay, because we're anti-parallel. So my oxygen will be here. You can do it, upside down pentagon. Cute. And then we have our phosphate group. So you can see how we took the same 3 structures, we just kind of flipped it and mirrored it. Ooh, that's my Fitbit. Not an Apple Watch, not yet. Garrett Fang, one day. Next we have an adenine. Adenine pairs with thymine. So let's draw our thymine here. Okay, make sure you have your oxygen pointing in the appropriate direction. Draw your pentagon and then your phosphate group. Cute. One more. Once again, if you're struggling, flip your notebook upside down. Thymine always pairs with adenine. Cat. Grand Theft Auto. And make sure you have your oxygen facing the appropriate direction. And then your phosphate group. Cute. Let's draw in our phosphodiester bonds. Remember, it's always between the phosphate group of 1 and the sugar of the next. Phosphate group of 1, sugar of the next. Cute. Now, I know you can already assume the directionality because it's going to be opposite, but it's nice to know how to figure that out without being given the opposing strand. So, using my rule, the oxygen, you always start clockwise. So clockwise from here is right here. So this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So my number 3 carbon is closer towards this side. So this would be my 3' end. And then my 5th carbon, so if we look at our last nucleotide, my 5th carbon would be here, closer to this end. So this would be my 5' end. Okay. The trick with the oxygen still holds true, right? The oxygens point towards the 5' ends. Cute. Let's draw in our bonds. So between our bases, we have hydrogen bonds. And we'll talk about why they are hydrogen bonds. So remember, cytosine and guanine will share 3 electrons. So we're going to have a triple bond here. We'll have a double bond between our adenine and thymine. So these are all hydrogen bonds. Cute. All right, so we kind of have 2 distinct areas here. We have these 2 ends where we have these repeating units, sugar, phosphate, sugar, phosphate, sugar, phosphate, phosphate, sugar, phosphate, sugar, phosphate, over and over again. Those are the 2 backbones. Those 2 structures are the same for every DNA strand you have. Your entire genome has that in common. The thing that makes us different is the sequence of our bases here, right? So like I might have black hair because my genome says CAT for my hair gene. Somebody else might have blonde hair because it says GAT for their hair gene, hair color gene. So depending on how you kind of change the sequence, it changes the proteins you get. And when you have a different protein, it can behave differently or it can make you look different. Cool. All right, that's it for structure. Come back and watch my DNA replication video. Bye.